Uh, Thank you very much for giving me this opportunity. And actually, I will start with a little bit of self-promotion because indeed we are very new here. So this is our space in February. And as of yesterday, we are able to use our new rooms. So this is how it looks like. And just again, very, very briefly, for those who are working here at the Institute or in the future considering working here in the Institute, we are setting up a lifestyle imaging facility. So we are very much looking forward to see everyone who has any kind of problems or has any kind of questions regarding imaging or image processing. And at the very least, I think we can try to give advice, but also we will be able to offer actually instruments, microscopes to use, as well as hardware and software tools for image processing. And also I take the opportunity to introduce my co-workers, Yasmin, who is a technician, and Antonio as a staff scientist, helping the facility, in particular with image processing and also microscope automation. So I will not talk more about the facility, but I will talk now about my other part of my job, which is to continue our research on oocyte meiosis and cytoskeletal dynamics in oocytes. So maybe I'm starting from very far, but well, I guess it's always good to start from the very basics. So you know very well that our world is organized from small elements, starting with atoms, larger and larger structures, and up to cells and make up organisms. So actually, I saw that many people show this kind of chart, and that's how they start their talk. But I actually think that one very important additional feature here is the diversity of elements. Maybe I played too much Lego when I was a child, but I think it's really the diversity of these elements that you can combine and build all different structures. I think that's what I find most exciting. And you could say what is very exciting is to put together proteins to large complexes, which many people here in the Institute do. But I would say our interest is at a bit higher level of organization. We would like to understand how these different molecular machines, like a nuclear pore complex or an actin nucleation complex or microtubules and microtubule regulators, can build all the diversity of different cells that come in really a wide variety of shapes and forms. And I think in cell biology in particular, somehow, this is not considered strongly enough, because